In this video, we're going to talk about cell division and the cell cycle, and more specifically, the control of the cell cycle. So this is how cells progress and make the decision to divide and continue with that decision. So first, let's look at the phases of the cell cycle. And the cell cycle is basically for cells that are going to divide. And let's start, for instance, in a cell that just came out of a cell division, so right out of cytokinesis. And that cell is a recently formed cell, and that cell would start in what we call the G1 cycle, or the growth 1 phase. And this is the phase in which cells will spend most of their life. Some cells never divide after a certain time. So for example, most neurons don't divide, and most muscle cells don't divide either. So those cells will go into what is called a G0 phase, in which they decided they're not going through cell division and they're going to spend the rest of their life in a G1 or G0 phase. Cells that will divide then will have to prepare to do cell division, and the first step in preparing for cell division is to duplicate your DNA. So you need to make a copy of that DNA so that you can give one to the daughter cell and keep the other one for yourself. And that is what is called the S phase. So it's S for synthesis, because this is when the synthesis of DNA is happening. So this is the S phase, where you're making a copy of the DNA. After you make a copy of the DNA, the next step is to get ready to actually divide. And then what you need to do is grow so that you have enough cytoplasm to divide in between the two cells. And you also need to have enough organelles to divide between the two cells. You need to have enough mitochondria, you need to have an endoplasmic reticulum large enough that you can split it in two, a Golgi apparatus that you can again split into two. All those things you need to have enough so that you can split into two cells. And that's what happens in the G2 phase. So the cell is growing and is making enough organelles so that they can separate them between the two cells. And once that happens, then the cell is ready to divide, which is the M phase for either mitosis or meiosis, depending on what type of cells. Cells in most parts of the body will do mitosis. This is cells that are in charge of growth or replacement. The only cells that would do meiosis are cells that are for reproduction. So those are cells that will become gametes. So cells that will form either sperm or eggs. All the other cells will do mitosis. And then the cycle will start again. So the cell will divide. We go through the phases of the division that it took, and then after cytokinesis you will have 2 daughter cells, or if you do meiosis you will have 4 daughter cells. So let's look at this question here. What stage in the cell cycle will this cell be? So if you look at this picture, this cell has 2 chromosomes. The large chromosomes that are homologous to each other, and then it has the small chromosomes, which are also the homologs. This chromosome has a blue gene, and this one has a pink gene, while this one has a yellow and this one has a green. So they don't have exactly the same information, but they code for the same type of genes. So these two will be homologous chromosomes, and these two will be homologous chromosomes. One will come from the mother, the other one from the father, and this one from the mother and one from the father. What you can see here is that the chromosomes have not duplicated yet. So they are single chromatids. This chromosome is a single chromatid, which means the cell has not duplicated its DNA. So it is before the synthesis phase. And clearly it has already divided because the chromosomes are single chromatids. So this cell will be on the G1 phase. A cell in the G2 phase will be after the synthesis, so you will expect that it has already doubled its DNA. So if it were in the G2 phase, it will need to have double DNA. And if it's on metaphase, then not only the chromosomes will be doubled, but they will also be attached to each other. And in anaphase, they will already be separated, but you will see that the cell has still twice as many chromosomes. So let's look at this other example. Now here you see that each chromosome has 2 sister chromatids. So that means each chromosome has twice as much DNA. This means that this cell already went through the synthesis phase and it already doubled its DNA. So this cell will have to be, it's not a G1, it has to be something after the S phase. If it were metaphase, again we said the homologous chromosomes will be facing each other, attached to each other, and they're not attached to each other. If it were anaphase, the sister chromatids had already separated, so that's not the case either. So it will have to be G2 phase, so right? The cell is ready to start cellular division, but it hasn't divided yet. Now I know if you're a purist, you're going to say, well, but these chromosomes are already condensed, and that's true. If the chromosomes are condensed, you would only see them in prophase, but here this is just for the sake of illustration. In the G2 phase, the chromosomes will not be condensed, they will be all spread out, and it will be impossible to see them as individual chromosomes, but for the sake of representation, we put them here as if they were condensed. So now let's look at the three main points in which a cell decides to commit to cell division, and it checks on itself and makes sure that it is ready to continue to the next step. So the first step starts in the G1 phase, and this is the phase where we said most cells spend their time, and if you look at a cell at any point in your body, more likely it is going to be at a G1 phase. This is when the cell is active and is doing whatever that cell is supposed to do. And that cell decides to divide. So the first thing it has to do in the G1S checkpoint is it has to check its own DNA, because you want to make sure that your DNA is good and it hasn't been compromised so that you can divide. You can give a copy of that to your daughter cell. So that will be the G1S checkpoint, where the cell checks its own DNA before it makes a copy of it. If everything is okay and the DNA is not damaged, then the cell will go ahead and start making a copy of the DNA, and that will be the S phase. So that's the synthesis phase. After the DNA has been copied, the next step, the cell starts to grow to prepare for division, so that's the G2 phase. And then another checkpoint will happen before the division, cellular division actually starts. In this point, the cell is checking that the DNA was properly replicated, that no mistakes were made, and it's also checking that it has enough energy and is of the right size so that it can divide into two. If all that is true, then the cell will go ahead into the next step and do cell division. Then, once cell division starts, there is a crucial point during the division in which the cell checks that everything is in order, and this is the spindle checkpoint. This happens during the metaphase part of cell division, and the cell checks that every chromosome is lined up at the center of the metaphase plate. This is very important because if a chromosome is not lined up there, that means that that chromosome is not going to divide properly. So one cell will get an extra copy of the chromosome and another cell would be lacking a chromosome. For example, that is how chromosomal disorders like Down syndrome happen. So if during cellular division one cell ends up with an extra chromosome and the other cell is lacking a chromosome, the one with the extra chromosome will, for instance, be having a trisomy, like what we see in Down syndrome. And all these points are very important because once you pass them, the mistake is going to be very serious or have detrimental consequences for the cell. So those are the key steps before you make a mistake that will be very costly. How are those cell cycles regulated? And the way it happens is by a combination of proteins. One which is an enzyme, the cell division promoting factor, and the other one is a cyclin which acts as a signaling protein. When those two are together, that gives the cell the signal that is the right time to divide. So those two act as a signal of cell division. And you can think of the need of having two proteins, kind of like what a bank has, to ensure that the safe is only open when two people who have different keys are present at the same time. So this adds extra safety to the cell to ensure that it's not dividing just because one person went out of control or because one protein went out of control. They both need to be present for the cell to divide. So these two proteins are a cyclin-dependent kinase. Cyclin-dependent means it needs to have a cyclin present in order for it to have its kinase activity or its phosphorylating activity. It also needs to be actively activated, so it needs to be phosphorylated also to be activated. So cell division will start only when you have enough cyclins and enough cyclin-dependent kinases, and the complex has been properly activated to promote division. This again ensures that the cell doesn't divide when it shouldn't. So there are many controls in cell division, because if cell division goes wrong, we end up with a disorder such as cancer. And this is very critical to keep cell division in check. Just to review, the points in which the cell is going to check whether it should commit to cell division: the G1S checkpoint, in which the cell checks that it has the signals to grow, that it is a cell that should commit to division, that it has a good quality of DNA and has a good size to go into synthesis of the DNA. Then the G2M checkpoint, which the cell will check that DNA was replicated properly and that the cell has enough organelles and enough of everything to split into two cells. And the third checkpoint is the metaphase, where the cell checks that the spindles are attached to every chromosome, and every chromosome is oriented in the metaphase plate so that the daughter cell will receive a copy of each chromosome. And this is a representation of that metaphase plate. So during metaphase, the chromosomes all get to arrange on the center of the cell and are attached to spindle fibers on both sides of the dividing cell. And this will ensure that each daughter cell will get one copy of each chromosome. So once the chromosomes split here, the sister chromatids are attached to each other by the kinetochores. Once the attachment breaks, there is no way for the cell to pair those up. So if they break and they end up in the wrong cell, there is no way to undo that process. That is why this step is so critical. So not only all those conditions need to be met, but a cell has also to be told to divide. And that's the role of growth factors. Growth factors are released, for example, when you exercise, you're releasing growth factors that tell your cells to divide, and that's why exercise increases your mass, but it also rejuvenates because it increases cell replacement. So those growth factors are telling the cells to divide, that there is a need to divide. A growth factor can sometimes be just when you take a cell out. For example, if you get a wound, then the absence of those cells would also stimulate cell divisions in the cells that remain. But any of these things can go out of control, and then it can cause uncontrollable cell division. And uncontrollable cell division is the beginning of cancer, or can lead to cancer. For example, if any path on this growth factor signaling pathway, where you receive the growth factor and then receptors relay that information onto the nucleus of the cell, where the proteins that signal cell division, such as the cyclins and cyclin-dependent kinases, start being made. If any of these elements here become stuck in the on position, so that it is telling the cell to divide even in the absence of growth factors, that is the beginning of cancer. And these elements here are very commonly linked to cancer. When any of those go wrong, the cell is likely to become cancerous. This type of genes are called proto-oncogenes. Proto-oncogenes means that they have the chance of becoming cancerous or oncogenes, but they're not there yet. So when they're normal, they're just proto-oncogenes. But if they go crazy, if they mutate, they get stuck in the on position, then they become oncogenes. So they're telling the cell constantly, divide, 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 even though the cell doesn't need to. And that results in cell division going out of control. There are other genes that do the opposite. They check for damage and they stop the cell from dividing. And if the damage is very serious, they might even ask the cell to do apoptosis, which is cell suicide. So the cell destroys itself before it starts dividing uncontrollably. So this type of genes are the ones that we have to think for when we don't develop cancer. They're called tumor suppressor genes, and they inhibit cell division if the DNA has been damaged and the cell is in no condition to divide. If you look at a cancer cell, it has been able to surpass this control. So most cancer cells do not have the signaling proteins. P53 is a common protein that detects DNA damage and then recruits other proteins to repair the damage. If the damage cannot be repaired, then it tells the cell to destroy itself. And like we said, for a cell to become cancerous, it has to be able to surpass this control mechanism. And often what happens is a tumor suppressor gene will be missing or it will be broken. So a mutation we have two copies of a tumor suppressor gene. If we inherit one that is already faulty, a damage on the second one is very likely to lead to cancer. And this is how some cancer types are hereditary, is usually because one of the tumor suppressor genes is already faulty. If you have two copies, then you have both of them will need to be broken for you to develop cancer. But if you only inherit a working one that works, then you're more likely to develop cancer. So just to compare proto-oncogenes and tumor suppressor genes, proto-oncogenes are usually related to growth, such as it can be the growth factor receptor, or it can be the RAS protein, or these are proteins that are involved in the signal transduction from the receptor protein to the nucleus of the cell, telling the cell to start dividing and start producing kinases for cell division. Tumor suppressor genes do the opposite. They check for the integrity of DNA and they check that the cell is in the right time to divide. And if it's not, then they will put the brakes on and they will stop cell division. This is why in many cases tumor suppressor genes are thought as the brakes in a car because they will stop cell division, while proto-oncogenes, if they become oncogenes, they can be understood as the gas pedal. So think of in a car, if your brakes fall, you can still control your gas pedal. Same thing, if your gas pedal gets stuck on, you still have your brakes. But if both of them fail, if you have your gas pedal stuck on and you have no brakes, the accident is imminent. And that's what happens with cancer. If you have a proto-oncogene that becomes an oncogene and starts dividing uncontrollably, and you lose your brakes from your tumor suppressor genes, then the result will be a cancer. And with that, we're finished with this video.